Right, I'm going to give you a bit of background first of all. I'm kind of going to split this into two parts. The first part is more of an introduction to me, how I got into science, and it's a bit of a weird pathway. And then I'm going to concentrate on apoptosis, but it is going to be fairly short. It's going to be brief. It's an incredibly complex area of biology, as most biology is, and I'm just going to skim the surface. More than anything, I'm aiming to just introduce you to an area you may not be aware of, and to just stimulate some interest in that, okay? Anyway, a bit of background about me. If you don't know me, I teach in the prep school, I'm a science teacher down there. I had a bit of a, it's weird being a teacher because at school I was a bad student. I wasn't very good, I didn't work basically. I could draw, I could do geography, that was about it. I didn't do anything else, muck around a bit. I probably shouldn't be saying this to you guys, but I wasn't interested in school at all. I started doing A-levels, I managed to get to do some A-levels, believe it or not. But throughout my school career, all I ever wanted to be was a Royal Marines officer. That's all that was going to ever happen. There was no other alternative for me. I hadn't even thought of one. So it was a bit of a shock when I'd done my potential officers course, and then I realised my eyesight was going, and in those days they wouldn't take you in if you needed corrective lenses. So for me, it was like, what do I do now? So I was at the point where I had to choose something rapidly. So I just applied to universities. I didn't know what I was going to do. I had no idea. I had no idea what subjects were available or anything. So I was stabbing around in the dark. And my favourite band at the time was the Stranglers. And I knew that the lead singer of the Stranglers, Hugh Cornwall, was doing a PhD before he joined up the band. And it was in biochemistry. So I thought, oh, that sounds kind of interesting. I'll apply to do that. And cut a long story short, that's what I ended up doing at university, okay? It wasn't until I got to university that I really got interested in science or in working in particular. And it really stimulated me because of the detail. Okay, so it's a bit of a weird background. It's not a standard background. It's probably not one you're probably used to. But that's how I got to university, came out with a biochemistry degree. I was going to do a PhD. I was looking at jobs as well, and I applied for this one with GlaxoSmithKline, as it is now. And it just happened that they were working on exactly the things I've been working on in my degree. So I got the job, basically. And it allowed me to basically be a student with lots of money, which was great. But it also allowed me to pursue a fascinating path through science. I was meant to be playing you a Twitter clip, but that's gone by the by, so we've got to knock that on the head. That's why I played a bit of music as well. Okay, so the aim of this is by the end of the session, hopefully I'll have given you a bit of insight into a specific area of cell biology, possibly stimulated one or two of you to pursue a career in science or down that pathway if you haven't thought about it already, and hopefully at least some of you will be awake at the end of it. Okay, right. Apoptosis, process of life through death. What on earth are we talking about? Okay, now the background slide is quite particular, and it might become a bit more apparent as we go through. Okay, so apoptosis is a process by which cells die. And we're going to get into that in a bit more detail later, but before we address that, we have to understand why there are certain processes in biology. And we, as humans, love grouping things. We put things into groups because it makes it easier to talk about. Now, for living things, we can group them in a number of different ways. But one of the ways we can group them is into unicellular versus multicellular. Now, unicellular, obviously the bacteria and the rest of it, are fairly simple. There are advantages of being unicellular, which you should be aware of, about diffusion of molecules and particles into the organisms, very simple. But there are drawbacks with it. And there are multicellular organisms. Now, multicellular organisms have massive challenges. Many of them, and you can probably see this number here, humans included, are made up of trillions of cells. And each of those cells has to work in unison for that organism to actually function as a unit. That's a massive challenge. That's 100 trillion cells in you working, hopefully, in unison to ensure the longevity, the survival, and the eventual proliferation of that species. Okay, so that's a massive challenge. Now I've chosen a picture here of a small nematode. It has very few cells. It's about 1,000 cells in the adult. And the reason I've chosen that picture is a lot of the initial research into a lot of developmental biology, and especially apoptosis, was carried out in C. elegans, Cenorhabditis elegans, okay, small nematode. It's very specific development of the cells, and it allowed biologists to study them and any knockout mutations within that organism, okay? Right, so multicellular organisms require complex strategy in order to survive. One of those things, and it is only one aspect of that, is the ability to selectively eliminate, get rid of unwanted cells. Now, those cells might be unwanted for a number of different reasons, and we'll get onto that in a minute, okay? But ultimately, the reason that that strategy has evolved is to maintain the organism's integrity and allow it to survive. Okay, cell death. This is apoptosis. It's programmed cell death. There are two ways in which cells can die. One is through insult, injury, chemical injury, physical injury. And the other one is actually through suicide. Now, that's a fairly aggressive word to use, but actually when we look at the mechanisms of it, that's exactly what the cells are doing. One of them is fairly random, it's not controlled, and it has issues. There are problems with the, this cell death. This one is specified, it's programmed, it goes down a very delineated pathway, and it helps the organism to survive. Okay, so cell injury, cells that die this way die by necrosis. And one of the problems with the necrosis is, because it's unregulated, the cells actually lyse, they spill their contents out, and that can have big problems to the surrounding tissues. It can cause inflammation, localized cellular tissue damage, and that's not what the actual organism needs. Cell suicide or apoptosis, programmed cell death, is very specified, it is controlled, and it doesn't result in lysis of the cell, importantly. Those cells that are programmed to die are actually swallowed up, phagocytosed, and eliminated in a very controlled manner. Okay? I've included this slide, it's not really that important, but what does an apoptosing cell look like? Normal cell, and we'll get onto what normal cells look like in a minute. When it apoptoses, it actually has a blebbing of the membrane, the DNA is broken down, cytoplasm shrinks. That is now identifiable by cells that are going to engulf it to be removed. And it's done so safely, there's no spillage of the internals. Okay, so that's what one looks like. Why should a cell commit suicide? There are thousands of reasons. This is literally not even skimming the surface. In virtually every biological process you can imagine on a cellular level or tissue organ level, apoptosis is involved. Okay? So examples are Apel's tail, interdigitation on fingers and feet, the menstrual cycle, so the losing of the inner lining of the uterus, formation of proper connections between neurons in the brain. That's a massive one, especially when you look at neurodegeneration later as well. Why do we need to destroy cells? So these are during development. Why might you need to destroy cells in an adult? If they're infected in a particular way, and the body can identify that they're infected, they need to be eliminated. Apoptosis is partly responsible for that. Cells of the immune system are undergoing cell clonal expansion and apoptosis all the time to regulate their numbers. DNA damage, crucial one, and we'll look at that in a bit in a minute, is about, obviously, that cell's not going to be any good to the organism as a whole, get rid of it, but in a controlled way. And the last thing is cancer, and we'll do a little bit on that about towards the end, okay? Right, developing a fetus in this case mainly involves cell proliferation in a very, very structured manner. Okay, you can see the head here. That's the remains of the neural tube, so you've got the brain and the spinal cord developing, and you've got limb buds. So the limb bud is literally a bud, that's what it looks like. But in order for that to develop into an arm, hand, fingers, digits, cells need to die. So rather than you having in the uterus that happening and you actually grow fingers, you have a big bundle of cells, and the ones in between your fingers actually die. But they're programmed to die, and it's a very specific method. It kind of looks like Homer Simpson, I think, but anyway. You can see the beginnings of the indigitation of the limb bud happening here, so obviously those are going to be the digits, and the cells in between will die. So a very basic schematic diagram of what's happening. Obviously in the frog, going from the sort of the morphogenesis between tadpole and frog, has to lose part of it as well as grow other parts in order to become the adult. All these cells die by apoptosis. They're programmed at a certain time to be gotten rid of. Interdigitation of the fingers, which I've just gone across. Now sometimes that will go wrong. It's fairly rare, but in a human sometimes it goes wrong. Not on a massive scale, and it's not debilitating really to the total organism's function. But you can see here that the interdigitation hasn't quite worked there, and it hasn't really worked there. But that's not a massive big deal. You can still survive with that. Okay, we're going to move on to cancer now. Now, cancer is obviously a massive issue in the press and everything. Everyone sort of has a bit of a flap about it. And on a personal level, it can be devastating. However, on a molecular level, subatomic level, it's fascinating. And well, I'm personally caught between the two, really, because I find this amazingly interesting. And actually, the more we can find out about the processes of cancer, obviously the greater the opportunity we have to intervene in a positive way to prevent or, in fact, reduce cancers in people who are suffering with it. Okay, there are two types of tumour, two main types of tumour. One of them, the first one, is benign. That basically means there's an uncontrolled proliferation of cells at a localized position in the organism, in the body, okay? That in itself is probably the result of a mutation in the DNA. So we've had one mutation, and that's caused this over-proliferation of cells. Normal cells won't do that. The normal cells here have what we call contact growth inhibition. So once they've grown to their normal size, they have contact between the cells, and that actually sends a signal to the cells to stop dividing. Quite often the benign ones, so that's the mutated one, lose that inhibition and they keep growing. Now ultimately that's not that serious. It usually results in a polyp or something that can be removed if it's detected. However, if that is allowed or goes undetected and another mutation happens, there is a possibility that it becomes malignant. Now malignancy depends on two things. It must be able to grow invasively, so through and into other tissues. But the main point is it has to undergo the process of metastasis, which means it moves from the area it originated from, travels through usually the vascular system to a secondary site. And that process is crucial in the development of cancer. It's not the only thing. There are other things involved. You can see that this is basically a tumour growing here, a malignant one, and it's got blood vessels in it. The process of angiogenesis, so basically the tumour's growing, but it needs a blood supply. So it actually stimulates the vascular cells to grow and provide it with all the goodies it needs through the blood system. So that's another area that I've worked on, trying to prevent angiogenesis, but I'm not going to go into that today. So, just to recap that bit, metastasis is a process by which cancer cells spread from the primary site to secondary site, and that's crucial in the mortality of cancer patients. Now, ironically, it's a massively inefficient process. It doesn't happen very often. It's a very rare event. And what I'm going to try and explain to you now is why it's a rare event and what happens when that event does occur. Okay? So, in order for metastasis for us to understand this translocation from the primary tumour site to the secondary tumour site, we have to look at the extracellular matrix. Now, I want to show of hands, who's heard of the extracellular matrix? Okay, fine. When we draw our pictures in biology, they're all nice and neat, and the cells are all organised and all lovely. It's not like that at all. Okay, it is in some tissues, especially if you're looking at a plant leaf or whatever, but in often mammals, it's a very, very dynamic environment. These cells are moving, and they're not just cells, they're actually attached to a scaffold. And this picture kind of illustrates it. This scaffold is called the extracellular matrix. It's just a bunch of proteins on which the cells attach themselves, and it allows them to crawl around to some extent. And we'll go into why that's so important in a minute. This is a cell, and all these things are this extracellular matrix, this scaffold on which the cells attach themselves. If we look at it as a picture, it's basically like this. This is obviously very oversimplified, but this is the extracellular matrix. All these ribbons and bands lying everywhere. They're just proteins. They don't really, well, they do a lot, but they're not that interesting per se. And in between them, we have lots of different cells. Okay, so fat cells, you've got white blood cells that are macrophages. Fibroblasts are actually the cells that produce the ECM, this extracellular matrix in the first place, but it's an incredibly dynamic situation in the body. Right, that's just another diagram showing you that actually, if you look at the blue things, the cells, so this is, I don't know what tissue this is, but it's just a standard tissue. A lot of the tissue is made up of the extracellular matrix rather than the cells. So it's something you're probably thinking, oh, that's a bit weird. It's something you're probably not used to because you have nice, neat diagrams in biology of what's going on. Okay, what does the ECM do, this extracellular matrix? This is a cell. This is the neighboring cell just off to the left. This is the nucleus. The extracellular matrix is down here. Again, this is very, very schematic, but hopefully it will give you an idea of what the role of the ECM is. Its role is multifunctional. But the crucial thing is that this cell has what we call transmembrane proteins. They're proteins that span from the inside of the cell to the outside of the cell. That's why they're called transmembrane. And if they are engaged in the correct way, they send through second messengers, which is a whole huge topic in itself, messages to the nucleus, not only to the nucleus, mainly to the nucleus, for the correct gene expression to happen, and that results in the correct proteins being made, and that signals the cell to do certain things. Now, it might be that it signals the cell to survive, and we'll go on to that in a minute, but it also can stimulate the cell to produce, if it's producing certain hormones or releasing things, it will stimulate them to do that correctly. The other thing it does is it gives the cell polarity. So you've probably, or should have, most of you should have heard of goblet cells. They're the mucous cells in your lung lining. They have to have polarity. They have to know which way to release the mucus, and that's away from the, so a goblet cell attaches to the ECM, and it releases its mucus out onto the ciliated cells in the lungs, okay? So cell polarity is crucial as well. This bit here is what I mentioned earlier. If you recall when I was talking about metastasis, most normal cells, when they grow in a wound, for instance, they will butt up against each other. And that inhibits the cell from dividing anymore. Okay, so that's basically a normal situation. Again, this is a slightly different diagram. Here's the ECM in this case up here, transmembrane domains. These are stuck onto the ECM. They are receiving signals saying, we're in the right place, guys. Everything's good. We're happy. Those signals are sent to the nucleus, either via this method or through growth factors. It's effectively the same thing. They're just little chemicals buzzing around. They occupy these receptors in the cell membrane. And this allows signals to be sent to the nucleus saying everything's fine. Now, because these signals are going from here to here, this blocks off cell death. Cell death is normal in every single cell in your body. It is the default pathway. And the only way it's prevented is by having positive signals sent to the nucleus saying, don't die, don't die, don't die. You're in a correct position, you're operating fine, carry on as normal. As soon as you remove this signal, so if this cell breaks away from the ECM, gets into the bloodstream and starts drifting off, in a normal cell, there is no signal then for apoptosis to not happen. So apoptosis happens, the cell dies. That is why metastasis is so inefficient, because you've got this default pathway saying, well, hey, hang on a minute, this isn't meant to be happening. I'm not receiving good signals. Let's go. Okay? However, in cancer cells, in the metastatic cells, this doesn't happen. The signal, even if it's removed, i.e. the cell is not in its correct position, it's drifting down the blood vessel, and it's going to be a problem potentially for the organism, apoptosis is bypassed. And that's usually due to mutation in the DNA. Right, that's pretty much it. I just want to finish, that's just a summary of what I've just gone through, so I'm not going to bore you with that. Just the last thing, it could be interesting if this works. Right, how many cells in me are going to die by apoptosis today? Let's just have some numbers, so it could be naught. 50,000, million? Could be 70. It's actually one of these numbers up here, but it's not quite right. Because it's 70 billion. So in a normal adult, every day, you're losing that many cells. Obviously, they're not just dying and you're not replacing them. They're getting rid of a continual turnover that allows this stasis of cell numbers. Okay, that's your lot for today. I hope you're still awake, some of you. Hopefully, I've given you a bit of insight into cell biology, actually how it is and how we can research it and the processes involved that are incredibly complicated, and I've only skimmed on the top of them. So thank you for your time.